Welcome to a walkthrough of WEGAN's statistical tools for analyzing ecology data. WEGAN is a web-based platform for analyzing ecology data that requires no coding knowledge. In this step-by-step tutorial, we will show users how to use statistical analysis tools such as chi-squared tests, t-tests, and fold change. Let's begin. Start by clicking the Statistics module from the home page to access statistical tests such as the chi-squared test, ANOVA, and fold change. Next, upload your dataset using the Choose File button. Alternatively, users can use sample datasets. For this example, we will select the Aravo sample dataset to load alpine plant distribution data. Selecting the Aravo dataset will also load the accompanying environmental data. Click Submit to proceed with your chosen data file. The data integrity check page will confirm whether the data is usable, such as checking for correct sample and variable labeling. See our data uploading tutorial for more details on how to upload data and manage errors. Here we see that our data has passed the integrity check. Scroll down to view a PCA overview of your data. Switch to the Heat Map tab to view a heat map overview of your data. The Data Editor tab can be used to make slight adjustments to the data. Here, we can change the data type, edit the data, and remove entries. Find more details on our data uploading tutorial. Once ready to continue, scroll down and click Proceed. On the normalization page, you can select the normalization methods appropriate for your dataset. There are 4 manual data transformation options to choose from. Click a radio button to select a methodology. Click Manual Normalization to apply your own data normalization configurations. A pop-up will show you variable data distribution plots comparing the effect of your selections. In this example, no normalization was applied. Switch to the Sample Overview tab to see similar plots for your samples. To make revisions to the normalization, click Go Back. Going back, we can alternatively choose Automatic Normalization to apply recommended normalization settings based on Pearson mode skewness. To move on from the normalization step, click Proceed. For ease of replication, this tutorial will continue with no normalizations. The second data integrity check page verifies data quality in the supplementary data. This page is for our environmental data. Here we see our supplementary data has passed the integrity check. Click Proceed to continue. After completing data pre-processing, you can select the type of statistical analysis to perform. We will begin with the chi-squared test. We have now reached the main analysis page. Here, we can adjust our test parameters and review our plot. The chi-squared test compares either 2 categorical variables or 2 species and 1 categorical variable to create a contingency table. Select an analysis type of either only categorical variables or 2 species and 1 categorical variable. Select whether or not to perform a continuity correction, which is applicable for a 2x2 contingency table. Based on your response to the first parameter, select your variables. For this example, we are selecting 1 categorical grouping variable and 2 species variables. Click Update to apply your changes. Click Customize Graph to change the appearance of your plot. Start by customizing the plot theme. Next, select a color palette for the plot bars. Select whether or not to display count labels. Choose where to position the legend. Finally, select whether or not to have a horizontal legend. Click Update to apply your changes. Scroll down to view your customized mosaic plot. In this plot, the 2 species variables are along the x-axis, while the categorical variable is along the y-axis. The area of each data bin corresponds to its proportion of the count data, similar to a pie chart. Click the Export button to download the mosaic plot. Select the Export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript formats. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select an export size of either full page or half page. Click Submit to generate the export file.
The download link will now update. Click it to open the export file in a new tab. Click the Results banner to expand a list of data files related to this sub-module. Click on a file to download it. Here we can view the detailed data table behind the mosaic plot. A pop-up shows us the statistics behind the mosaic plot in table format. Moving forward, we can navigate to another analysis method from the left-hand menu. Note that fold change, t-test, and volcano plot analyses can only be applied to two-group data. For the following examples, we will switch to the BCI dataset as it is two-group data. We will apply no normalizations. The fold change analysis compares the absolute value of change between two group means. This is calculated as a ratio between the two group means before column-wise normalization is applied. First, select the grouping variable. For this dataset, there is only one option. The analysis type can either be paired or unpaired. For this example, the analysis type cannot be changed. When performing paired analysis, the program first counts the number of pairs with consistent change above the FC threshold. If this number exceeds the count threshold, the variable will be considered significant. Input a fold change threshold. This cutoff must be greater than 1. The default is 2.0. Next, select a comparison type. This tells the program which direction to compare the groups in. In practice, changing this option flips the negative and positive sides of the plot. If applicable, input the significant count threshold percentage as needed. Click Update to apply your changes. Scroll down to view the fold change results. Remember to export your plot. Under the Results banner, we can download the analysis data and view a detailed data table of the significant results. The detailed data table shows the statistics for the significant results of the analysis. Click on the Here link to download the table. Moving on, we will continue to the t-test submodule. In a t-test, each dependent variable is individually assessed against the grouping or independent variable. Select the grouping variable to perform the analysis against. Select if the group variance is equal or unequal. Select whether to perform the parametric t-test or non-parametric Wilcoxon rank sum test as appropriate. Input a false discovery rate threshold as needed. This only applies if the analysis is paired. Click Update to apply your changes. Scroll down to view the t-test results. Here, we see one significant data point with red coloration. Clicking on the point at 152 and 460 opens a box plot showing its analysis results in detail. Click Export to download this plot. This downloads only the t-test plot, not any adjacent box plots. Under the Results banner, we can download the analysis results and can view the detailed data table. As there was only one significant result based on our test parameters, we only see one row in the detailed results table. Click the Here link to download the table. We can now proceed to the volcano plot analysis. The volcano plot combines fold change and t-test analyses. A volcano plot is a type of scatter plot used to visually display the results of high-throughput data analysis, particularly in differential expression studies. Adjust the variables as needed. Select whether to label the plot when exporting. For the p-value threshold, specify if it is for the raw or the FDR-adjusted p-values. Click Update to apply your changes. Scroll down to view the volcano plot. This plot shows the statistical significance as a negative log 10 of p-value along the y-axis. The x-axis shows the magnitude of log 2 fold change. Significant data points, if present, will be colored red. We can view the details of a data point by clicking on it. For example, clicking on the point at approximately 2 and 0.75 opens the box plot on the right. Click Export to download this plot. The export will only include the volcano plot. Under the Results banner, we can download the analysis results or view the detailed results in a table. However, since our plot shows no significant results, these files will be empty. Click on the detailed data table to see more. In the detailed data table, there is no data. This is because there were no significant results in our analysis, so no data is recorded in the output table. We will now proceed to the ANOVA analysis method. For the following analyses, we will switch back to the Aravo dataset with no normalizations applied. ANOVA, or analysis of variance, determines if there are significant differences between the means of 3 or more groups. It compares the within-group and between-group variabilities to identify if at least one group mean is significantly different. On this page, users can either perform one-way ANOVA or the non-parametric Kruskal-Wallis test. The false discovery rate, or FDR correction, is employed to ensure that the overall false positive rate is appropriately controlled. Input an FDR cutoff. The default is 0.05. Select a grouping variable. Each dependent variable will be assessed against the grouping variable. Select a method to use in post-hoc analysis. Finally, select whether this will be a parametric or non-parametric ANOVA test. Note that post-hoc analysis will only be performed with parametric ANOVA. Click Update to apply your changes. Scroll down to view the ANOVA plot. Click on any point to view a detailed box plot on the right-hand side. For example, clicking on the point at 57 and 2.07 opens a detailed box plot view of this data point. Click Export to download the plot. This export will only include the ANOVA plot and not any adjacent box plot. Under the Results banner, we can download the data file related to the ANOVA analysis and view a detailed table of the plot results. Click to view the detailed table. Here, we can see a detailed description of the post-hoc tests and significance results. To download this table, click the Here link. We will now proceed to the Pattern Hunter submodule. Pattern Hunter analysis is a method used to identify and analyze patterns within datasets. It uses statistical techniques to detect regularities, trends, or anomalies. Correlation analysis can be performed either against a given feature or against a given pattern. First, select the categorical grouping variable. Next, select the pattern to search for in the data. There are 3 pattern searching options. The first option is to select a variable as the pattern and specify that variable. The second option is when the pattern is specified as a series of numbers separated by a dash. Each number corresponds to the expected expression pattern in the corresponding group. The number to label allocation of the groups is given as the first item. The last option is to define a custom pattern profile. After defining the pattern, choose a method for calculating distances. Click Update to apply your changes. Scroll down to view the plot. Remember to export the plot. Download the analysis results from under the Results banner. Finally, for assistance with any of the PLSDA methodologies, refer to our clustering and classification tutorial. Once finished with all analyses, proceed to the Download option from the navigation tree. Click Results to expand the list of files available for download. A list of all data files from this analysis session will open. Click on any file to download. The download.zip file contains all listed files. Download the WEGAN Docker file to recreate the WEGAN coding environment. Download the RSource file to get the exact R script used during your analysis session. After exporting your results, click Exit to return to the home page. We hope you found this tutorial on the Statistics module helpful.